So, good afternoon, everyone. Thank you for attending this second webinar of the ECPGR Grant Scheme Activities series. And we will have today a webinar offered by the Allium Working Group, specifically related to the Safe Ali Div project. This was carried out between 2017 and 2019 with a budget of 29,000 euro. And it was focused on safeguarding potato onion in the northern region and the Baltic area. And this project was very interesting because it touched a peculiar type of germplasm, referring to this potato onion that is not known to everyone, but is an Allium cepa varietà aggregatum in taxonomic terms. The presenters will confirm or not what I'm saying. And there is a very interesting traditional type of growing, especially in the Baltic area, but not only. So we will hear from about this on-farm conservation aspects and also ex situ conservation aspects of this crop. We have two presenters today, Helena Stavelikova from the Crop Research Institute of Olomouc. She's the chair of the Allium Working Group, and she will be followed by Dainis Rungis from the Latvian State Forest Research Institute in Salaspils in Latvia, and he's also the national coordinator for Latvia. So I will open now the presentation. Should last in total about half an hour, then we will have questions and you will be allowed to speak by us. So Helena, go ahead. You can start your presentation.
It means that I put the screen hot, yes?
Yeah, yes, please share your presentation.
Yeah. Good afternoon, everybody.
We cannot see it yet. We cannot see the presentation yet.
We cannot see the presentation.
No. Yes, now.
Now, yes.
Go ahead. Yes.
Now, yes.
Yes, go ahead, Helena. Many thanks.
Yeah. Good afternoon, everybody. My name is Helena Stavelikova, and I'm chair of Allium Working Group. And I would like to present the project, the safeguarding of a potato onion, Allium cepa aggregatum group, and garlic, Allium sativum, crops diversity in the North Europe and Baltic region, in acronym Safe Ali Div. The project was financed but under the 4 calls of the ECPGR activity grant scheme in phase 9. The presentation has 2 parts, the general part with me and the part about the molecular analysis with Dr. Dainis Rungis. The project has a duration 24 months, and the name of the aim of the project was the promotive active on the farm conservation of European allium accessions, and the second, the genetic analysis undertaken within the project complemented the existing molecular markers analysis of Nordic potato onion that was recently carried out in Finland and Sweden, with analysis of additional material from partner countries, in order to reach a better understanding of existing genetic diversity in collections. And the project has two sub-objectives: to investigate the situation in Europe in allium crops cultivation, both commercial and home garden scale. And the second, to clarify the scale of potato, onion, and garlic cultivation as part of the biocultural heritage. The project has had 12 partners, 11 funded partners, and 1 self-funded partners were participate in the project. The colleague from Czech Republic, me, from Estonia, Prit Portman, from Finland, Tehri Suala Ahalfors, Ligalebse from Latvia, Daugulo from Lithuania, Hriskik from Netherlands, Ingun Molet-Wagen from Norway, Matteo Leino from Sweden, Kuli Anama from Estonia, Dainis Rungis from Latvia, and Smiljana Goreta Ban from Croatia. Therefore, the project was focused to region Scandinavia and Baltic region. The project partners were mainly from this region, but we had 3 expansions. Hriskik from Netherlands as the expert on evaluating DNA analysis, Smiljana Gorena Ban from Croatia, because we wanted to have material from the diametrically different geographic area in the project for comparison, and me from Czech Republic because the Czech shallot collection is specific in that it has many materials from Finland, Norway, and Sweden. The collections of other partners represent only their national project. Sorry, national material, not a project. During the project, we had 2 meetings. The first meeting was held in Tallinn, Estonia, from 11 to 12 July 2017. During the first meeting, the partners presented the situation in allium crops cultivation and conservation in their countries, with emphasis on potato, onion, shallot, allium cepa aggregatum group, and garlic, allium sativum. The second, the group visited potato onion fields in the Lake Peipus area, where traditional cultivation is based of heterogeneous seed propagated material, in contrast to the customary vegetative propagation in other countries via bulbs. It was agreed that the different propagation methods influence the genetic and the morphological heterogeneity of accessions as well as their phytosanitary status. At the tree, it was agreed to perform also morphological characterization of the some accessions which were to send for molecular analysis to Latvia State Forest Research Institute. For morphological characterization, we used the descriptor list allium subspecies by IPGRI 2001. And of course, for the clarify the existing situation in potato onion conservation and diversity. Before this meeting, the project partner received and completed the questionnaire, providing the following information on the 3 allium species: garlic, allium sativum, potato allium, potato onion shallot, and onion allium cepa. The number of accessions, type of collection, the field or seeds collection, type of conservation, it's mean ex situ, in situ, on farm, in vitro, source of new accessions, for example, collection mission, seeds company, breeders, and etc. Number of description accessions, number of accessions with chemical analysis, and number of accessions with DNA analysis, safety duplication, cooperation with other GBank in accessions exchange, and current project related to particular species, and other genetic resources related activities. We receive to the information about the commercial production for each species was also included in the questionnaire, and the number of national varieties, harvest area, production, import, and home garden scale. I think it was very interesting that the question was sent to us partners project as all members of ALIA working group. We received 23 answers, and over of all answers in the table. The table is very, very large. It has been divided into parts, and I choose only the part table with the data about the potato, onion, allium cepa aggregatum, because normally the table was very, very long, and here is some photo from the excursion in the region and Lake Peipus, where we visited 2 local growers with this material. The second meeting was held in Olomouc, Czech Republic, from 17 to 18 April 2018, and at the first we discussed the task from the first meeting and that fulfillment, and the second Dainis Rungis from the Latvia State Forest Research Institute presented preliminary results of the molecular analysis. In the second part of this presentation, Dainis present the results of molecular analysis, and the third we agreed that we use for morphological description disease characters, folium colors, folium attitude, bolting, bolt skin color, with scale brown, yellow, and red, bulb flesh color, bulb hurting, bulb shape, and harvest. And here we used the free scale, flat, round, and elongated. And the partners of project visited the G-Bank of vegetable and special crops in Olomouc, where they could observe a collection of garlic and shallot. Here are some photos from the excursion on the field collection, and one photo from the Olomouc town hall. And here we met with vice major of town Olomouc. I think it was interesting for the all partners. During the meetings, we discussed the practical question of maintenance on vegetatively propagated allium, and I think it was very good for all, because the changing the practical information is very, very important, and personally contact is more better than only contact via mail, telephone, and other system. It's my part of a presentation, and thank you very much for your attention. And now Dainis present the results of the molecular analysis. Thank you. Hello.
Yes, thank you, Helena. Can you stop sharing, and Dainis will continue with his now.
Okay, so can you see the presentation?
Yes.
Yes.
Yes. Okay. So yeah. So thank you to Helena for a nice introduction and overview. So I will be presenting the results from the genetic analyses that Helena mentioned. So basically, this is all this work was published in 2020, I think, in a paper in the Genetic Resources and Crop Evolution, with all the co-authors that Helena already listed and mentioned. So, okay, so again, this is just a little bit of an introduction. So as Lorenzo mentioned, the potato onions are maybe not another very major crop, but they're cultivated in home gardens and in the sort of Nordic Baltic region, as well as southeastern Europe. And again, so they're becoming more interest in terms of this sort of food movement and so on. So again, Helena touched on this. So they're vegetatively propagated, except for in near Lake Peipsi, well, there was a visit. And again, so these have been very valuable genetic resources. The Estonian ones and some varieties have been developed from these Lake Peipsi onions. So where did potato onions, well, and how they moved throughout Europe? So there was some, this is from Eric's master's thesis, I guess, yeah, master's, in terms of this was looking at sort of west to east or north movement of germplasm and so on, but there's not much information. The other aspect that we wanted to, or our hypothesis was looking at movement of this germplasm from the east, from Russia, but yeah, basically we could not get any samples from Russia itself, and so therefore I'll touch on this later, but we couldn't really expand on this idea. So what did we do within this activity? So we had a total of 264 accessions from all the partners listed. Some, again, Helena mentioned most of the accessions from each country were local accessions from each country, except for the Czech Republic, which had quite a number of accessions from safety duplicates and others from the Nordic countries. In addition, then we had two accessions, which were these seed propagated Estonian accessions. Which were also included. So we basically just took bulk DNA from 2 individuals, and for this the seed propagated accessions, we took 9 individuals from each accession. So he's genotyped them with the 11 markers, SSR markers, and these markers had also been used previously for analysis of potato onion in Sweden, which was published by Matti Laino. And also for Finnish, which, as far as I know, or at least at that time, was not published yet. So that in any case, we could potentially also compare and combine these data sets. So on to the results. First one's more of the housekeeping side of it. So because we knew that there were some safety duplications in the collection, we could check this. In most cases, it was okay. In other cases, we found some where they didn't match up. And again, some genotype or some accessions had identical genotypes, but most of them were from the same collection. And they were also collected, had similar names, or collected from similar regions and so on. So we're not sure if they're not actually. So there's two possibilities. One's that these accessions may in fact be genetically identical, and they just annotated with different names, or they, as is written, could represent cases of historical movement of the germplasm. The other option, or the other possibility, is that the SSR markers that we used were not able to differentiate accessions, but we couldn't really, without a more thorough phenotypic and genetic analysis, we couldn't rule one or the other out. So at least it's a start to in terms of tidying up these collections. So maybe on to the more interesting part about the genetic diversity and differentiation. So in terms of the diversity, what was interesting was that we found that the genetic diversity within the 2 Estonian seed propagated accessions was almost the same as in all the others. So we had 18 individuals from the 2 seed propagated accessions, and 258 individuals from the vegetatively propagated. Yeah, and as you can see, the genetic diversity indices are quite similar between the 2 groups, irrespective of their sizes. And again, all the 18 seed propagated individuals were genetically unique as well, had unique genotypes. So we were half joking that we could actually just maintain these 2 seed propagated accessions, and we could maintain almost the same amount of genetic diversity as in all the entire accessions. But so, and then in terms of genetic differentiation, so when we look at compare, so here we took out, so this was all the previous was all the accessions. Here we took out. And from each country were only accessions which were actually from originated from that country, as far as we know. So in terms of the Czech collection, we took out all the known accessions which were safety duplicates, which were and had an origin of outside of Czechoslovakia, sorry, Czech Republic. Yeah, and so as you can see here. I'm not sure if you can see my pointer, but the main we have here at the bottom is the Nordic, Norway, Sweden, Finland, and Czech collections. In the middle, we have the Estonian, which also includes these seed propagated accessions, and then more differentiated, sort of the Latvian and Lithuanian collections, and the Croatian collections, which was the most differentiated. So just to sort of sum up what we found, so the results are consistent with previous molecular analyses. I mentioned there was the paper published by Matti looking at Norwegian and Swedish potato, onions, and Finnish, I think, as well. So here the conclusion was that the Norwegian and Swedish collections were quite close, with Finnish being more differentiated. So we found this as well. And in that paper, it was suggested that the differentiation of the Finnish potato onion may have been due to the sort of influence of germplasm from Eastern Europe, including Russia. And there was also an hypothesis that some of the Latvian potato onion germplasm could also have been originated from Russia and east of Latvia and the Baltic states. However, we did try to obtain some material from the Vavilov Institute, which was not successful during the study. Also, there were some Russian accessions in the Czech collection, but I think most of these were from Siberia and not so very distant, and so not really relevant for looking at the germplasm movement. So therefore, we couldn't sort of test this hypothesis or confirm or refute this about this northeastern, but it still remains open, since the sort of relative differentiation of the Latvian and Lithuanian collections, and we do see this sort of gradation of Estonian, Finnish, and then I guess the Swedish and Norwegian and Czech, which could be more of a Western European origin. So briefly, that's it for me. Thank you for your attention. Thanks to Helena for as the working group leader, as well as for coordinating the activity. Thank you to Liga as the Latvian member of the Allium working group and for assisting in these analyses, and of course to ECPGR for funding this activity. Thank you.
Thank you, Danis and Helena, for these presentations illustrating this very peculiar area of European germplasm. So I know there is a lot more in what happened in this project because of the interactions that you had also with farmers, etc., and that may be developed perhaps in a few questions here. But I would open the floor to the audience if anyone wants to comment or make specific questions for the presenters. You can raise your hand and I am supposed to unmute you if you wish to speak, to ask a question. So while people think, I can ask you or anyone that participated in the project, may be present in the audience, if you can tell us something about the growing situation in I mean the on-farm situation for this crop, whether it is still valid, and whether your project has enabled some promotion of the on-farm conservation or increased knowledge in these countries about these crops, and whether that was necessary, maybe not. Is it anyone wants to answer, Helena or Danis or someone else? Helen, we cannot hear you. Oh, Helena, are you speaking or no? Dinis, can you say something? Because I cannot hear Helena.
Yeah, yeah, I can't hear. I think I'm not sure if Helen is. But yeah, no, I'd leave that question to the other members.
Okay.
Yeah, there's also Liga. Sorry, there's Liga and Wisław who have their hands up. Maybe they want to say something, Lorenzo.
Yes, I'll give first to Liga. Let me.
Also Eric.
Also Eric, yes. Okay, Liga was a member of this activity, so go ahead, Liga.
Hello. Thank you, Helena and Dinis, for nice presenting of our project. And I just have very short comment on growing of potato onion. I would say that it's not grown in commercial scale at the moment in Latvia, but as Dinis or Helena, I don't remember who, one of them mentioned that it's kind of home gardener's crop, let's say, and it's more like a heritage crop. But still, I would like to say that there is rising slowly, there is rising interest from chefs, from restaurants to have those potato onions because of their rather high pungency. At least those genotypes which are coming from Latvian and probably also Lithuanian collections, they are rather pungent with a high pungency, and they are giving a special taste for food. So chefs in restaurants, they are interested. So I think probably this crop will become a niche product for small scale farmers or for organic farmers probably. But I see the potential of the crop.
Okay, thank you very much, Liga, for this clarification. And so Wisław is now with their hand raised. So Wisław, I'm trying to unmute you. Go ahead.
Do you hear me?
Yes.
Oh, thank you. So first of all, congratulations on the project. Very interesting result. But my question is focused on the nomenclature. You use 3 different names for the onion: potato onion, shallot, and aggregatum. Is there any difference between shallot and potato onion, or not, or it is synonym?
Okay, thank you for your question, Wisław. Is there anyone who wants to answer from the presenters?
Yeah, understand me?
Yes.
Hello?
Go ahead,
Yes.
Helena. Yes.
For the identification, we use the taxonomical from the from the books Allium Crop Science by Chaim Rabinovich, and we used this scale for this project. Because takhle Allium cepa aggregatum group and the botanical names of the crop groups, and other name used in the literature is the Allium ascalonicum, Shallot Allium cepa variatum aggregatum, Allium cepa variatum ascalonicum, and in English name the shallot potato onion and multiplicaire onion, and we used this taxonomical system.
Okay, thank you. Because shallot is a well-known cultivated plant, and is even registered in, as a cultivar, and on the common catalog there is several varieties of shallot or Ashalanicum. So it's not only the species which is typical for the small farms or new. It's not in fact in some regions not new crop.
I think in the minority species is little problem. It's a very local name exists. For example, in the Czech Republic, shallot or potato onion has around 10 local names, and maybe it's the problem that every country has used a special local name, and sometimes it's result a little bit confusion situation, because where we prepared this project, we had very rich discussion about this, and finally we chose this possibility, as I say at the first time.
Yes, I agree with you.
Yes.
We have the same situation that when we are collecting, we have asked for not for potato onion, but for the family onion, familiar onion, because the shape of the group of small bulbs. Yes, that's true. But in case of potato onion, I heard that this is a special type of onion which is used during which was used during boiling potatoes to improve the taste of potatoes. And for this reason is the name potato onion. But it's, I heard it from old manuscripts, and also in these manuscripts, shallot and potato onion are different things. So I wonder if you examine your material on this aspect, that maybe it's some variety or race or forma of the aggregatum group, from the aggregatum group.
Wisław, I can witness that the taxonomic issue was debated also in this group, and very difficult to find a unanimous idea, but probably it is something that maybe the Allium group could consider to elaborate a bit on the use of these names, if they find the courage to do that.
Yes, I know that
I share my experience from the collecting.
Yes, thank you, Wisław. That was a very nice information that you gave. I would like to let the floor to Eric, who has the hand raised since quite some time. Let's hear what he has to say. Eric, you are self-muted, it seems. So I'm not able to unmute you. Try to unmute yourself because. Okay,
Now,
go
can you
ahead.
hear me now? Yeah.
Yes,
Yeah.
go ahead.
Yeah. Thanks a lot for a nice presentation. A little bit on the same issue, I think. I think that these common names maybe shall be like treated like part of the big diversity of this crop in Europe, because it's hard to get some common ground, I guess, because it's so much different aspects of these in different countries. And in fact, I got an article published just today in Genetic Resources and Crop Evolution about introduction of potato, onions, and shallots to Sweden, where I also are writing a bit about these different ways of distinguishing between shallots and potato onions in England, France, and Germany in historic times. But I guess this is just a glimpse of the diversity in this kind of area as well, because in Sweden we know that the both names, both shallots and potato onions, the names has been used in different regions for the same plant material, and this genetic work that has been done has helped us to confirm that this is actually the same genetic plant material. It's not clonically identical, but it's not possible to separate between shallots and potato onions, at least in a Swedish context. Again, thank you a lot for the presentation. I can also add that there's a huge interest in this crop as well in Sweden for home growers, and these different land races are sold to home growers a lot and very popular. Thank you.
Thank you, Eric, for this useful addition, confirming that the taxonomic issues is relative in some cases. Helena, you are raising your hand, or?
So I have
Yes?
a small, remarks to material from Russia. Of course, I had a very rich corresponding with the colleague from Cycle Petersburg about the possibility sent to us some samples of Russia material, but finally it was impossible because we had only Russia material from a Czech collection, but we tried.
Yes, it is clear, and I think it's increasingly difficult. Thank you, Helena. Any other question from the floor, or suggestions for Eric? Your hand, oh, Jelka. Eric, I suppose your hand is from before, so I give the floor to Jelka.
Yes, yes.
From Slovenia.
Do you hear me?
Yes, go ahead.
Okay. Thank you also from my side to Helena and Rangis and for the group for this interesting project. I am not a specialist in onion, so it was also interesting for me to know this on the taxonomy, because we also in Slovenia we used to grow, and are still growing shallot, but I was also wondering already at the meeting we had in Poland about the difference between shallot and onion. So yeah, it's a little bit now. It has been answered, so it's difficult. So we also grow, although we don't have any shallots or potato onion in the gene bank. However, my question would be: you mentioned, Helena, at the beginning that you covered also the aspects of on-farm conservation. So can you maybe tell something more on this or the project partners? Thank you.
Because we are in the questionnaire, we asked for all allium species which the members of allium working group had in their collection. And in this presentation, I choose only shallot, only potato on onion. And in this in a shallot is not exist in the on-farm conservation, but some species also allium in maybe Finland, I don't know exactly in this moment, use the farm conservation, but I must see on the original big table. Yes, but where are asked in the questionnaire, we put the possibility to possibility the system of conservation. It means sits on field conservation, ex situ, in situ, on farm, on cryopreservation, on in vitro, and the number of member of group wrote what is actually for their collection. But this data came from 2017 years. It means it's the 7 years ago. It means that during this period situation can be choose. And I think that it's good every time on the beginning of the new phase of ACPG, the new phase, to send the same questionnaire to all members of area working group, that chair or vice chair can to use the actual data, because I know from me that the situation is not stagnant, but is continue and comes some change, and I think it's the good during 4 and 5 years to receive new data about conservation of allium species in the GBank, because it's very good to have this data when we prepare some project and etc. It's good to have this this information on the table.
Thank you, Helena. And I would like to remind you that it's also possible to look at the report of this activity on the website, and the report includes also some public awareness products that were published during this project. Also, they are in local languages. Maybe not everyone can read Finnish or Estonian, but they're also visually interesting. And I would like to throw a challenge to the allium working group, if you can, considering what was the interest that this difference between shallot and potato onion continues to raise. If you could develop a very simple short paper for genetic resources, our journal, with whatever is available to differentiate or not shallot from potato onion. That would be a delightful exercise, I think.
Maybe to moment. Yeah, it's the good idea. Maybe it's the good topic for discussion with Eric and Matti from Sweden, because Matti has an interesting article about it, but I think it's the mainly the top discussion for taxonomical specialists, because it's a very special problematic.
Yes, of course, but there is also the
maybe
local taxonomy.
I ask other partners of this project what they mean about it.
Yes, exactly. Thank you, Helena. So we are reaching the end of this hour. If there is any other relevant questions, we have 2 minutes, but I do not see any hands raised. In which case, I would thank everyone for presenting, you presenters, and for attending. I would like to remind you that we will try to continue this series of webinars with other activities, showcasing them in the next months, and we are looking for volunteers. We will find volunteers if necessary, but spontaneous volunteers would be really preferred. And this webinar has been recorded, so you will be able to watch it again. And with this, I would like to invite you to the next webinar that will be announced shortly on the same platform. Thank you, everyone, from the CPGR Secretariat, and goodbye.
Yeah, goodbye.
Bye.
Thank you. Bye-bye.